Okay, we go 3, 2, 1. Hey, yeah, very good. Hi, everybody. It's a real pleasure to be here. The class finally starts. So I'm very excited to be here. And I'll start with one thing that I wanted to put on record about the mask. So as you know, the mask is not mandatory at all at the university from this moment. The way I see the mask is that this is a device that protects the people who are not wearing it. Okay, so of course it protects me because I have it now, but it's mostly a good protection for people who are in front of me. So that's why I keep it, is because as simple as it sounds, I value your safety more than my comfort. Okay, it's not because I want to make a statement that you have to wear a mask or something like this. That said, there's a distance of more than 2 meters between the pulpit and the first lines of chairs, and if that's okay with everybody in the room, I'm going to remove my mask during the class because it's going to be much easier for you to understand me. It's also going to be much easier for people online to understand me as well. And I think that this is relatively safe in the sense that there's a huge distance between you and I. Okay, so it's just something that I wanted to make sure that everybody's fine with it. So hi, that's me. I look like that without the mask. It's a pleasure to see you and to meet you. And the reason I'm excited is because this today is exactly the thing I wanted to do all my life. Yes, no kidding. So that's my first class giving statistics classes in a university, in an amphitheater, in an auditorium, and that's really one of the main things I hope to be able to do in my life. So that's such a privilege to be here and to be able to do this with you guys today. So the first thing we're going to do is to go as rapidly as possible through a couple of slides that explain some things about this course so that you're a little bit oriented. And then we're going to dash forward as fast as possible on the questions that we have to answer today. And again, the logic is because we want to, get those questions answered so that you guys can have the grade as highest as possible. That said, it's an interactive class, okay? Which means interrupt me anytime, online or in person. We're here to discuss, we're here to talk about the things we don't understand. So just because we want to be focused and make progress doesn't mean that you guys should not interrupt me, okay? So let's go with the presentation. Is this shared online? I'm not quite sure that the screen is shared. Can you guys see my screen with your instructors right now? Yes, okay, that's good. So that's me here. My name is Guillaume Filion, and RTA is Sarah Saloum. So my name here is a little bit hard to pronounce. That's a French name that's pronounced Guillaume, and my family name is Filion, much easier. So just a few words to introduce me. So I've been doing academic job for a long time already, and I started my studies in 1998. My first university was in Lyon here in 98. Then in 2000, I did 1 year in Montreal. It was super nice to go to the Concordia University. Then from 2003, I did a master's and a PhD in Paris. After this, I did a postdoc in Amsterdam, starting 2008. In 2012, I became a group leader, a principal investigator. So I was a scientist, but not a teacher. So worked in Barcelona for my first academic job as an independent scientist from 2012. I worked 9 years there, and then finally I got a position in 2020 at the University of Toronto. Timing was exactly excellent because I came at the same time as COVID-19. So this is now my first day in the real world teaching in an auditorium, even if I've been here for 2 years. I guess this is the same for many of you. You've been here at least 1 year, maybe more. And this is one of the first time you have this experience, even though you've been here some time. So that's me in a nutshell. Other things about my work, so is that most of my activity is research, it's not teaching, okay? So consider me as a researcher who's doing some teaching. And in the lab, what we're doing is that we're working on chromatin and HIV biology. I don't have much time to explain what chromatin is, but basically that's the molecules that are around DNA and that influence DNA. And we're really interested in questions about what this has to do with HIV and with viruses in general, because they have to integrate inside the genome. So we're really asking whether that affects the life cycle of the virus. And half of my time I also spend on more theoretical questions. and I particularly develop models of deep learning. Everybody, I suppose, knows what deep learning is by now, and probabilistic programming, which is something that we're not going to see in this class too much, but maybe a little bit. So at a more personal level, what do I like to do when I don't teach and when I don't do research? Well, I like blogging, and I like blogging about statistics. This is one of my hobbies. And I like to blog also about the way we apply statistics to biology, etc. And I called it Life for Statistical Sciences, because that's really the way I think about my blog. Also, I like science fiction and I like math. I do an hour of math every day just for sport because I love it. Science fiction because it's fun. So a few words about Sarah. She would present herself, but she's online, so I'm going to just introduce her very rapidly. So between 2018 and 2022, she was at the University of Toronto. She was an undergraduate. So she just graduated recently. From 2021 to 2022, she came to the lab as a research assistant. She's also starting right now as a research assistant. She's doing a master's in cells and systems biology. Anybody who's interested in prolonging their studies with a master's, maybe you can talk to Sarah. She's very friendly. She can explain to you what the perspectives are if you want to be a grad student one day. And in the future, she'd like to pursue a PhD in computational biology, hopefully in my lab. We'll see about this. Other things about Sarah is that she speaks 3 languages fluently: Arabic, English, and French. So don't be shy. You can practice any of these 3 languages with her. She's got a gym obsession. She trains 5 to 6 days a week. She's very serious about it. And she loves reading textbooks and papers for fun, aren't we all? So that's about it for your teachers, very rapidly. And now, you may wonder, what is an introduction to computational biology about? It could be many things. And one thing I really wanted to focus on is the statistical aspect of computational biology. So mostly we'll be studying statistics more than traditional computational biology. Why will we study statistics? Is because it's the new thing for you. That's the most important. This is really what develops a certain frame of mind. It's not just a set of skills, a set of techniques, it's a way of thinking. And so one thing that I like to compare life with is this TV series, which is The X-Files. So when I was a teenager, even a young teenager, that was the hot thing on TV, was The X-Files. There was nothing else at the time, so everybody was watching The X-Files. And Fox Mulder, the hero, had this poster in the back of his office. It looked exactly like this, with a UFO, and it said, "I want to believe." And so, of course, everybody was, it was such a superhero, and we all loved it. But growing up a little bit, I thought, it's really fascinating how "I want to believe" describes in so few words the way we think normally. The reason I say what I say is not because they're true, it's because I want to believe they're true. Right? So when we're not thinking in a particular way, that's the way we think. We say things because we want to believe they're true, not because they're true or not because of anything else. So that's what I consider is the normal way of thinking. And that way of thinking brings us somewhere, but as scientists, that cannot be a good research for truth, right? You don't really want to say what you say just because you want to believe it's true. As scientists, we got to have something better. What could be better? Well, it's hard. That's really damn hard to do better than "I want to believe." That's really hard. And that's what statistics can help you with. And that's what statistics is good for. Right? It's not the magic bullet. It's not perfect, but it's already an improvement over this. So statistics, I define it as the way of changing your mind. Okay, the science of changing your mind. I have no problem changing my mind. You will see I change it all the time. And there is a reason why I change my mind. There's a logic to it. And this logic is the logic of science, the logic of statistics. So in this course, we will develop a coherent way of thinking about problems in biology. Our conclusions are not so important as the argument we will put forward to reach these conclusions. Okay, so whatever we learn in this class, I don't want you to focus on what we learned. I want to focus on the way we learned it. Okay, we're going to just show a step-by-step method to approach problems in a quantitative way. And so that's really my hope for this class. I hope that we can do that together. All right, that's all I wanted to say about the course. I know that you have plenty of questions about the exams, the coefficients of the exams, etc. But if you're okay with it, we'll talk about this as we're going. Okay, unless there's really an urgent question, like I really cannot wait to ask you this, then ask it now. Otherwise, we're going to get started right now with the basically with the questions, and I'll be answering, and you'll understand exactly how things work. It's going to be very repetitive after 1 or 2 classes. Any urgent questions? Not in the room. Urgent questions online? Oh, there's one there. Oh, sorry. Yeah.
Sorry, I don't have this course in further since I have human biology instead.
You have what?
I have human biology instead of computation biology in further. Who do I contact about that? Registrar's office?
Yeah, you have to talk to the registrar's office with this. But so you would like to study human biology or computational?
No. This is the course I'm supposed to be in.
I see.
I have human biology in further or something.
I see. Okay. Very good. So for those online, it was a personal question. Okay, so if we're done with this, then go ahead and take your PDF here, BIOB21_1 with random shufflings. And we're going to start with the questions directly. So I have my OR session here, and it's not exactly the same as you guys will have. There's no color on mine, and the reason why I'm using this one is because I can actually increase the size so that it's going to be easier for people to read it online and in the class. Okay, so here if I type, I hope that most of you will be able to read the letters that I'm typing here. Okay, so go ahead, start R. In principle, you know what I mean when I say start R, and take the first the first PDF, the one that corresponds to today's class, September 6th, and we get started with the problem. Maybe you've looked at it already, and you've seen that I would like to talk in this particular class about COVID-19 vaccines. So that's a little bit, undiplomatic of me to start with something that is such a point of disagreement. And I did this exactly on purpose. Okay, I wanted to start our classes on computational biology with exactly that question of the COVID-19 vaccine. And the reason is not because I want you to believe that they're either safe or unsafe or efficient or not efficient. What I would like you is to ask yourself what you really think about that. Okay, I don't have any particular conclusion about that. Okay, I think that anyway, most of you must be vaccinated because I think it was mandatory last year, and very few people have managed to go through. I know that some people didn't like the fact that they were forced to vaccinate, and that's something that is not a discussion about today. So if it's the case, right, I would like you to actually keep your doubts. This is exactly what this is good for. And if you have no doubts, I would like you to try and remember when you had doubts, because I would like to review this from the perspective of computational biology and from statistics, not from the point of view of biology, not from the point of view of policy making. All right, so this is an effort that I'm asking everybody to do. Let's roll back to a time when we were undecided and review what the arguments were of the people who published the scientific evidence. These are not my arguments. These are probably not your arguments either, but I want you to know those arguments and to understand how they work so that you can comment on them. After this, you can disagree with it. Of course, you're more than welcome to disagree. I value the differences of opinions and points of view. But before you disagree, you have to know what you disagree with, okay? What you disagree against, in a sense. So that's why I chose something that's a bit debatable for the first class, but I know that's something that has huge consequences for the world, and that's the main point, is that these decisions that were made just shaped the life of millions, hundreds of millions of people. And these decisions were made on statistics. And that's why I would like to review the statistics with you today. Okay? If that is a problem for you and you think that this is unpolitically correct, etc., well, you're very free to raise your voice and to talk to me openly or in private. I'll take the criticism. But I hope that for now you can kind of roll back to a time when you had doubts and then kind of rediscover this question from the point of view of statistics. So let's go. At the onset of the COVID-19 epidemic, multiple projects were initiated to develop vaccines against SARS-CoV-2. Some of these projects were based on recent technologies to trigger the immune system with an mRNA. The efficiency of such mRNA vaccines had been tested as cancer treatments, but not for immunization campaigns against infectious diseases. We will review the efficiency data of the clinical trial for the mRNA-1273 vaccine produced by the company Moderna. Okay, so today is about the Moderna vaccine, not the Pfizer vaccine. The vaccine was designed on January 2020, within 24 hours of the publication of the SARS-CoV-2 genome. The phase 1 trial started on March 16, 2020, the phase 2 clinical trial on May 29, 2020, and the phase 3, the one that we're talking about today, on July 27, 2020. The initial results of the phase 3 trial were published in early February 2021. So that here is the work that is based on the result of this phase 3 trial. We will use R to analyze the outcome and to quantify, sorry about the typo, our confidence in the results in the efficiency of the vaccine. So randomness and the pseudo-random generator, we're going to kind of warm up a little bit and to make sure that everybody's on the same page and gets kind of fluent with using R. So question 1, start R, type the command sample x equals 10 and press enter to obtain the same result as shown below. So here we go. This one's fairly easy because it tells you what to do. I have a bit of an issue here. Maybe I can make that a bit smaller. Okay, you anyway have the document, so you can read it. And here, sample x equals 10. Okay, you guys can do it on your computer. Do we get the same result? Obviously, no. Okay. So there's a note, the greater than symbol in front of the command is called the prompt. Okay, this thing here that I'm highlighting, that's called the prompt. When you type, you don't type the prompt when you're on the R session. Okay, why do we not get the same result? What's happening? Anyone? The numbers are generated randomly. Yes, what did you want to say? Yes, exactly. The numbers are random. Okay, that's the reason why they're different. And that was the whole point of the video. It was to explain the difference between a function that is deterministic, in which you always get the same result, and a function that's random, that uses the random number generator. So what is the random number generator good for? To generate randomness. And we will see why we need to generate randomness is because of the uncertainties about everything in life. So we need models for randomness. Okay, so the first question is very easy. Do you obtain the same result as shown below? No. Whatever your computer is, you don't obtain the same result. So what is the chance that someone just out of random would get exactly the same numbers as here? They're slim to none. That's why I'm comfortable that no one just by chance will get that. Question 2, now use the command set seed 1 to 3 before calling sample x equals 10. Okay, so set.seed 1 to 3. And then we repeat the same thing. So just one thing for you who are watching, you can see how fast I tap when I do this. So how do I do this? I'm just pressing the up arrow on my keyboard. When I press up arrow, that regenerates the previous commands that I tapped. So that's something I'm going to be doing all the time to save, to go really fast. So I'm pressing the up arrow 2 times, and that brings me to sample x equals 10 again without me having to retype everything. It's very, very handy to save a lot of time. I do this all the time without telling you, okay? But keep the habit of doing that. Whenever you made a typo or whenever you want to reuse the command, etc., regenerate the previous command with the up arrow, fix your typo, or just press enter to run it again. Okay, so now we get 3, 10, 2, 8, etc. And magic, we get the same numbers as in the PDF, right? And if you're doing it right now, I'm quite sure that you get the same numbers. If anyone at that point, if anyone does this command and does not get the same numbers, please tell me. Okay, that's really, really, really important that we understand why you would not get the same numbers. Everything depends on you getting exactly the same numbers as me for the Quarkus pages. Okay, so if there is a bug, or if there's something voodoo happening on your computer, then I need to know it so that at least we can fix it. Okay, so far so good? So the number 1 to 3 in the previous example is called a random seed. There's nothing special about 1 to 3 other than it is a popular choice and that it is the number I usually use. I, Guillaume, use that number all the time. Okay, 1 to 3 is just like any number, but you'll see online when people use set seed, they will often use 1 to 3. You have 1, 1, 2, and 1, 2, 3. These are the very common choices. I like 1, 2, 3. Okay, so what happens inside? Why do we get the same numbers now? Anyone online, anyone in the room? Yes, go ahead. Yes, so I'm resetting the state of the random variable, and then from that point, we set it to state 1, 2, 3. Okay, it's actually a very complicated thing, a random number generator. It's really complicated, but every number corresponds to a state. You can just put a label on it. This is state 1, 2, 3. We all put our random generator in that same state, and from then it's like a wheel that turns. You know, we all make one step forward, but it's exactly the same step forward. That's exactly this. Like we have reset or set the state of the random generator to whatever is arbitrary. We take 1, 2, 3, and that's as good as any number. As long as we agree on the number we use, then it's fine. Question 3, try using different seeds starting from 1, 2, etc., which is the smallest seed such that the 10th value in the output of sample x equals 10 is equal to 10. Yes? Yes. Absolutely. So the question is, if we set a seed, then is it deterministic after we set the seed, right? And the answer is yes, it is. It's completely deterministic. Okay, and this is where we come into something that is colliding with our intuition very strongly. How can this be random if it's deterministic, in the sense that there is no uncertainty whatsoever, okay, about the process, in the sense that I do the result first, you do them after this, but you knew what you would get. You know, they were on the screen first, so that is deterministic. How can this be random if it's deterministic? The answer is quite simple: is it's not random, it's deterministic. That's why we call it pseudo-random. Okay, pseudo-random means not random at all. Okay, so how can we then consider that this is still random if I just told you that this is deterministic? Well, we have to kind of discuss what we mean by random. And this is where it's really complicated. And we're not going to be doing a lot of philosophy in this class, but still every now and then it's good to think about what we mean when we say random or probable or something like this. Okay, and there was one question in particular at the quiz that people found hard, which is, once I said this 1, 2, 3, is this biased? And many people have said, yes, from now this is biased because I set the random seed, but the process is not biased. So what's the answer? Well, it depends on what we call random. So random intuitively means unpredictable. Okay, so it means that something is random if I cannot predict it, but from the mathematical point of view, it's got nothing to do with unpredictable. It's gonna mean measurable. Okay, so if half of the numbers are produced, let's say I'm using the random number generator to produce numbers between 0 and 1. Okay, and I set the seed to 1, 2, 3, and then I start producing numbers. If half of the numbers I produce are between 0 and 1/2, and the other half are between 1/2 and 1, then that's good enough from the point of view of probabilities. Okay, the fact that everything that we can measure on them looks like the frequency of appearance is the correct one, then that's good. The fact that they were predictable is irrelevant. So very philosophical. I'm sorry if I lost some of you or all of you on these things. So the answer is, it is deterministic. We call it pseudo-random because it's very handy. And from all practical purposes, it is indistinguishable from random. Okay, it looks like random. And if you want, the way you can think about it is that I can, if I know the seed and you don't, for you it's random. Okay, so these things would be completely random for you, and if it's hard and it causes you problem to say no, this is deterministic, etc., think of a way where I would be running all these things without even showing you the random seed I use. Then for me they would be deterministic, for you they'd be random, and everything would be fine on your side. Okay, it's a bit of a mental gymnastics, and I'm sorry again if that causes a lot of problem for you. How can this be random if it's deterministic? Don't lose sleep over it. Let's move forward. If it really prevents you from moving forward in a couple of hours or a couple of days, talk to me. Let's go and talk at the you have office hours as well. But otherwise, you'll see it won't really prevent us from making progress. So try to ignore it for now until you kind of get a feel for what it means. Any other questions before we move on? I know this one is hard, and that's why I wanted to put this out of the way in the first class. The pseudo-random numbers are deterministic. Yes. All right. Question 3. So I read it already, so let's go ahead. So we start using different seeds, starting from 1. So here, the way I do it is I do set seed 1, okay? And then I repeat sample x equals 10. So I get a different set of numbers as the previous time, of course. And the question is that we have to find a case where we're going to get a 10 here, that the last value is going to be a 10. Okay, so obviously not this one. So how do we do this? Press arrow, up arrow 2 times. Let's try seed 2. Do it again. Not this one. Okay. So here we go. We'll try 3. Most was the 9. One more. Still not. Still no luck. Okay, 5. Still not. Getting repetitive. Okay, bingo. Got it. Okay, so in principle, everybody who has done this, you've seen that on your computer, when you use set seed 6 here, then you get exactly the same output as me. And this is the first, starting from 1, where we get the output here at 10 at the 10th position. Okay, so this exercise is just to practice a little bit the fact that you could use different random seeds to reproduce different random situations as usual. And it's just to try and use different seeds for the same problem. There's no particular lesson beyond that on this one. All right, question 4. You will use the random seed 1 to 3 to answer some questions on Quarkus. Here is a practice round to demonstrate how it works. When you see the indication in red that you have to use seed 1 to 3 and concatenate, if you're using Quarkus, as in this question, run the code as shown below with set seed 1 to 3 and concatenate all the numbers and paste them in the answer box. Okay, so you will have to put all these things on Quarkus. Okay, and to make sure that we can play with random and we can still make sure that you get the correct answer, then the way to do this is to use specific random seeds, and we'll be using 1 to 3 all the time. So here we go. You do this example. So set seed 1 to 3, always the same, and then sample x equals 5. We get 5 numbers in that particular order. Okay, so 3, 2, 5, 4, 1. So the answer then on Quarkus is the number 32,541. Okay, no space, no nothing. You go on Quarkus and you put 3, 2, 5, 4, 1. So this one is a free answer. That's the first time, just to demonstrate how this works, because you'll be doing this a lot. Okay, to use set seed to make sure that you get exactly the same random number as I do. And on Quarkus, then it will validate that you have exactly the right numbers. Okay, the chance that you get exactly the right numbers by doing the wrong things is pretty much 0. So that will ensure that if you've got the right answer, then you have done the right thing. Okay, so this one was just to make sure that everybody's on the same page. Here, there's nothing to do. It's only for people who are filling it on Quarkus as we go. Then the answer would be 32541. All right, is that clear for everybody, this thing? I think you'll get used to it very soon anyway. Okay, so let's get started with the bit more complicated things then. Random draws with sample. So the R symbol sample represents a function. To make this clear, I will write it as sample, round brackets, 3 little dots between the round brackets, reminding ourselves that it should always be used with some arguments that are passed between brackets. Okay, if I type sample here like this, what do I get? I get gibberish. I get actually the definition of the function, which is something that's unintelligible for me. So when I call a function without the round brackets, then it's not useful. I don't get something that's interesting for me. So I always have to call the function with the round brackets. Okay, and I have to put something here inside at the place where I put the 3 dots. Okay, so one can access the documentation about OR functions such as sample with the commands help sample and question mark sample. Okay, so that's something that was on the video, and I put it on the very first video because you'll be doing this a lot. Whenever you're lost, of course you're going to use Google, you're going to use Stack Overflow a lot, but before you do this, you can make a habit of checking the internal documentation of R, and you're going to get information that you do not get on Stack Overflow or on Google, like very precise information about this. Okay, so using the documentation, if necessary, modify the call sample x equals 10 so as to draw 5 numbers at random between 1 and 10 without replacement instead of the default 10. Okay, so what do I mean here? I say that sample x equals 10 gives me 10 numbers. Okay, they're all different from each other. It's the first 10 numbers and they're scrambled. Now, what I would like is to get 5 numbers from these 10. Okay? I don't want all of them. And I don't want to reuse the same number twice. I don't want that in the output there is the number 1 2 times and the number 2 2 times. Okay, so any idea? Anyone knows how I can do this? How should I modify this thing here to sample 5 numbers between 1 and 10? Yes. Size equals 5. Excellent. Okay, if I just put x equals 5 here, that's not the solution because I get a random shuffling of the first 5 numbers. So here, I think that was in the video, size equals 5. Okay, so I have to put now that I want x equals 10. X is the number of integers that I'm going to sample from, and size is the size of the sample I get at the end. Okay, so you see 9, 3, 8 to 7, it's 5 numbers indeed. There's no repeat. We've seen each number only once, and so that's something that's reproducible. We don't get the same number twice ever when we do that. Okay, so what do I mean by using the documentation if necessary? So either you knew that, you remembered it, or you've seen it somewhere else, or someone told you, or you don't. In that case, there's no way you can guess the right answer. So the way to do this is question mark sample. And then that shows something that looks like that. If you're using the other version of R, the one that I'm using on the videos, then it's going to open a new window for it. Okay, but basically you're going to see something with the same information, more colors maybe, but the most important thing is that, okay, is that usage shows you all the available parameters for a function. And this is where you can see that there's size, there's replace, and there's prop, and there's nothing else. You're not allowed to pass anything else than these 4 items to sample. And if you don't know what size is, you go to argument, and then there it is, a non-negative integer giving the number of items to choose. Okay, it's hard to understand, but by kind of playing around a little bit, then you answer your questions this way. So if you're using the same version of R as me, which is on the terminal, to get out of the help is to press the letter Q, but I don't think that anybody's using that version. Okay, so that's what I meant by using the documentation if necessary, modify the call, blah, blah, blah. So, okay, what's the answer on Quarkus? Then we have to go back to our favorite seed, okay? Set seed 1, 2, 3, sample equal, x equals 10, size equals 5. And so that's it. That's the answer here, okay? That's 3, 10, 2, 8, 6 without space between. Like you put that as your answer on Quarkus, that is the answer. Okay? So far, so good? So there were some questions on the chat. Okay, add size equals 5, amazing. All right, that's good. All right. So we're still warming up here, just making sure that we understand how to answer the questions on Quarkus. Things still don't look like biological, but soon they will. Question 6, further modify the call of question 5 so as to draw 5 numbers at random with replacement. Okay, so any idea, anyone? What should I do in that particular call here to allow the same number to be drawn multiple times? You're here. Yeah, comma, replace equals true. Okay, write true in capital letters or understands only true in capital letters. You cannot write true in small letters or with a capital T. Okay, if you're particularly lazy, you can write just a capital T. That will work as well, but I don't recommend it. Okay? I recommend writing true in capital letters every time, and this is why I will write true in capital letters every time. I'm going to shortcut it with T. Okay, so here we go, replace equals true, and that's it. Okay, there's no repetition in that particular example, but if I keep drawing, you can see that I got a 999 here. So obviously repeats are allowed. They're not obligatory, but they're allowed with replace equals true. All right, so here we go. I said seed 1, 2, 3 again, replace equals true. So that's the answer on Quercus. Okay, it's that number here, 331026. That is the correct answer on Quercus. All right, so that was just to make sure that everybody's comfortable with sampling, and we'll be sampling a lot in this class. Everything is based on sampling. Creating and combining vectors. The OR function C dot dot dot, because it's a function, is used to concatenate multiple values into what is called a vector. For instance, below is the call to create a vector with the first 6 prime numbers. Okay, so if I do this thing here, and I paste it here, 2, 3, 5, 7, 11, 13, these are the prime numbers, the first prime numbers. Okay, you can see that I get that. There's this little 1 here between brackets, and for now it's not very useful because our outputs are pretty compact. So there's only one number like this, and it says that the number that's here is the first element of the vector. That's what it means. Okay, so that's one example in which I can create here a vector that contains anything I like. I just have to put things inside this C, that stands for concatenate, that creates a vector with these numbers. This is a vector with 6 elements, or a vector of length 6. The number 1 square brackets in front of the output indicates that the index of the first value, which is 2 in this case, is 1. Okay, like it's the first value is 1. It is just a way to keep track of the indices when the vectors are long. So not really useful now, but at some point it will be useful. You can ignore it for now, but this will become useful at some point. Question 7, use the function sample to pick 5 numbers at random from the first 6 prime numbers without replacement. Make sure the prime numbers are passed in ascending order as above. Okay, so here this one is kind of just to practice a little bit. So here instead of x equals 10, what am I going to put? Go ahead. Yeah. Yeah, exactly. Okay, so if I do it without the C here, it's not going to like it. Okay, if I just do that here, it's really going to, so in your case, going to be red, and it says you're using arguments 11 and 13, so it really misunderstands what you're saying. Makes no sense at all. Okay, what's worse is that if you do that, it's maybe gonna work. Thanks God, it doesn't work. Okay, so anyway, the point is that you need to wrap this up inside C for concatenate. And now that's working. Now it's producing some numbers. Okay, so by default here, sample produces all the numbers without replacement. Just puts them in a different order. So what do they want? Pick 5 numbers at random from the first 6 prime numbers. Okay, so how do I pick only 5 out of these 6? Yeah, exactly. Okay, size equals 5. Okay, now we get only 5 numbers, and by default replace equals true, so is false. So if I really want to be verbose, I can say replace equals false here, but that wasn't really the case. Okay, so whether you do this with replace equals false or without replace equals false is the same thing. By default, it does not replace. We're never going to see the same numbers appearing twice in the sample. Okay, so that was it. Now I just need to get the right answer. As usual, I use set seed 1 to 3. I plug in the right call here, and I get 5, 13, 3, 7, 11. That's great. Just to make sure that I did not say anything that was incorrect. If I say replace equals false here, you can see that I get exactly the same result. Okay, so saying replace equals false is the same thing as not saying anything at all. Right? And the answer on Quarkus is 5133711. Okay, like one number, everything concatenated, no space in between. All right, so the purpose of this exercise is to see that with sample, I can put whatever I want. Okay, I can just put a couple of numbers and ask R, now draw things at random, do whatever you want with it, give me a random sample of something that I'm presenting to you. And the way I do this is with this little construct C. The function rep is used to repeat. Okay, it stands for repeat, REP. The same value or the same vector multiple times. For instance, below is the call to repeat 30 times the value 0. Okay, so if I do rep x equals 0 times equals 20, so then obviously it should be 30 here. Sorry about the mismatch. So I think it's pretty obvious that this will repeat 20 times, not 30. Right? So here is the call that you need to type to repeat the same value 30 times. Okay, x is the value you want to repeat, times is the number of times you want it to be repeated. Okay, now use the function rep to repeat the pair 0, 1 50 times. It's not really clear why we're doing this now. We're just doing a little bit, warm-up exercise, flexing our muscles, but that will be useful just from the next paragraph here. Okay, repeat the pair 0, 1 50 times. So what should I write here in the call? How do I change it so that I can repeat not just the same value here, but the vector? So here, what do I write, you say? C01, right? Like this, and times equals 50, of course. Okay, and then I get something extremely interesting, 0, 1, 0, 1, 0, 1, etc. Okay, so that's what we wanted. We have repeated the whole vector here 50 times in a row. What is the length of that vector? Okay, so here that starts to be interesting. Is it 50? No, obviously not. Okay, how do I know? It's because here there's 75 here. So that 0 is in position 75. Okay, so this 0 is in position 1, that's the first value. This 1 here is in position 38, and that 0 here is position 75. So 75, 6, 7, 8, etc. You can see that this one is going to be number 100. So the length is 100, it's not 50. Okay, so the way you do it is either you do it this way, like you say, this is 75, and then I'm going to count until the end, till this is here 100, or you get a little bit more science. You use the function length. Oh, almost. And you get 100. Okay, so these numbers here on the left, when the output is short, they're really useless, they're redundant. But when the output is longer than a row, okay, then they start to be a little bit useful because you can get an idea of how long the output is, and you can know that, for example, okay, this is here position 76. That's fairly easy to find, given that this one is 75 here. Okay, so the answer is it's length 100. It's not length 50. Okay, like because you repeat the whole vector 50 times, the vector was length 2. So that's the way rep works. It's length, so times is not the output length, it's really the number of times you repeat the same pattern. All right? Okay, so on the chat, yes, length is 100, exactly. Very good. All right, randomized clinical trials. So I think now is a good time to have a little break, maybe a 5-minute break, to just like get your eyes off the screen. Maybe go for a tiny walk if you want, or something like this. So we're going to get back shortly after 6, if it's okay with you. Okay, so a short break of 5-6 minutes at most, and I'm going to pause the recording in the meantime. And we're back. So while people are going back to their chairs, so one thing I'd like to mention is that this is an experimental course. So that's the first year it's given, and so far this is an elective course. You don't need it for anything. Nobody needs it. It's not a prerequisite and doesn't lead to anything. So I'm given carte blanche to try a new way of teaching statistics and to teach computational biology. So it's going to be a little bit unconventional this way. And my point of view is that to understand the fundamental ideas, we don't need the math because we got computers. Most of these things were invented around 1900 to 1950. A lot of it was done in this time, way before we had access to computers. And I think it's time to revisit the way we understand and we teach these things with computers. So my idea is we can replace all the hardcore mathematical equations by computers, but that's a guess. Maybe it's not possible, maybe it doesn't work this way. So that's a very experimental course, and I hope that you're okay with going on board for the experiment. If you're not, then no problem. If you want to, say like, not for me, I want a normal class with normal math, etc. I think the best class for this is a psychology statistics class. That seems to be the best at the moment. But the course is going to be like this all the time. Okay, there's not going to be any math in it. There's going to be a lot, though, of simulations using the computers, because I think that we can understand much better how things work if we can make them concrete thanks to computers. This is the first time this course is offered, maybe the last, depending on the feedback we get. Really, I'm very, I mean it. I really value feedback. If anything you really don't like or you think is not working for the way you understand things, tell me and I'll change it. Also, I prepared the classes only a couple of classes ahead so that I can reorient, change a little bit if things are really not working at all on your side. You need to tell me, and I'll make sure that we can actually get these concepts on everyone's mind. So now let's go back to where we were. I wish we had a computational bio major program at UTSC. Yeah, that's kind of that's the agenda, is that we start with this course, then a minor, then a major. It's going to take a couple of years. So you're the lucky ones. You're the ones in the experimental side, and your feedback will have huge impact if this project of computational biology minor and then major goes through. Okay, so randomized clinical trials, and this is where we actually start now for real. A clinical trial is an investigative protocol to determine the efficiency and the safety of a given medical treatment. It doesn't have to be vaccines. Patients are enrolled in a cohort that is monitored by physicians who test the treatment and eventually determine if it is effective. Clinical trials are organized in phases that serve different purposes. Phase 3 is usually the last, where the purpose is to test the treatment on large samples in order to validate it on all the representative groups of the population, and otherwise identify groups of people that should not receive the treatment. Okay, so phases 1 and 2 is to test that the treatment is not killing people just all the time or something like this. So when they reach phase 3, it has already there's already evidence that the product is effective, but you want to test like really, are there some groups of people who really don't benefit from the treatment? So this is why you take as big a sample as possible. And in this case, they had something like 15,000 already. So that's supposed to cover a good representation of the population that you target. To establish a baseline for comparisons, only some patients of the cohort receive the treatment, while others receive a placebo. Okay, and this is like really the innovative nature of medicine, of the Western medicine, is this idea of comparing something to a placebo, which also has ethics side to it. Okay, half of the people we don't treat. And so how acceptable is that? That's not something we're going to discuss now. That's just the way things work, and you have to know that they work this way. It's not possible to have a treatment that is accepted by the FDA or any organization if you don't test it against a placebo. Okay, so when I was younger, I thought, well, it's kind of unfair to not treat half of the people. Now that I get older, I'm thinking, well, we don't know which half is the lucky one. You know, if the treatment happens to have severe consequences, the ones who had the placebo are actually the lucky ones. To establish, okay, so a placebo is a neutral molecule, usually water, that is packaged like the treatment so that the patients believe that they receive the treatment. That's the whole point. They must believe that this is the treatment. They must not know that they have the placebo, because there are huge psychological effects on the side of the patient, but also on the side of the doctor. A clinical trial is randomized if the decision to give the treatment or the placebo is random. Okay, that's what is a randomized clinical trial. This is considered best practice to avoid treatment biases. Example, when physicians decide to give an aggressive treatment to the patients who are the most likely to tolerate it. So there are examples of this. One of them was the chloroquine, if you remember. So like something that's really hard to tolerate, so people give it to the patients who are the most likely to resist it. But if they are the most likely to resist it, they're also the ones who are the healthiest, the ones who are the least affected by the diseases. And so when you don't randomize and you come to the conclusion that chloroquine works, it's also because it was given to patients who are the least likely to experience negative side effects or primary effects even. Okay, you want to avoid that, basically, this kind of situation. The clinical trial is blinded if the physicians do not know whether they administer the treatment or the placebo. Okay, like in that case, it's called double-blinded. People never know that they receive a placebo, but either the doctors know or they don't know. If they don't know, it's called blinded. This is considered best practice to avoid interpretation biases. Example, when physicians run more diagnoses on patients who receive the treatment. Okay, if they want to test the safety of a treatment, and they know that this person has received the treatment, and they look for every possible thing that can go wrong, then they will find more things that go wrong if they run more tests. Okay, so then you will have a bias between what you find for people who have received the placebo, people who have received the vaccine. So you want to avoid that as well. You want it to be blinded. The phase 3 clinical trial of the mRNA-1723 vaccine was observer blinded, meaning that the person doing the injection knew whether it was the treatment or the vaccine, but no one else did. Okay, only one person knew in the Moderna trial whether this was the placebo or the vaccine, and that's the person who did the injection. But after this person just like wrote it down on a secret piece of paper and sent it away so that nobody could know. So the patient did not know that they received placebo or the vaccine, and everybody around in the facility or the nurses, they also did not know. In the words of the authors, once the injection was completed, only trial staff who were unaware of treatment assignments performed assessment and interacted with the participants. Okay, like in that sense, there is no bias. That the person who knew just went away and was never seen again. The logic of this protocol is that if the treatment is effective, the patients who receive the treatment should be healthier than those who receive the placebo. But the results are never all or none. So we need to interpret them with a critical mind if we want to evaluate our confidence in the outcome. Okay, and that very last sentence is exactly the reason why we need randomness. It's because whenever you do a clinical trial, it's never the case that 100% of the people who have the treatment feel good and 100% of the people who have the placebo feel bad. It's never like that. It's always something in between. And this is why you need randomness in order to understand whether what you see is it for real or is it not working very well? So let's go back now to reenacting this clinical trial. Question 9, let us replicate the Moderna clinical trial and draw a random sample of patients from the cohort. The authors write, the trial enrolled 30,420 volunteers who were randomly assigned in a 1-to-1 ratio to receive either vaccine or placebo. 15,210 participants in each group. Okay, you understand how this works, right? Like they randomize everybody who comes, say, I am a volunteer, I want to get the vaccine. So sure, go in line. And then they randomize them into either the group that will receive the placebo and the group that will receive the real vaccine. And so they got exactly the same numbers in both cases, because for every person that they put in one, they put another one in the other branch. So they're called the placebo branch or the treatment branch. So assign the number 0 to patients who received the placebo and number 1 to patients who received the vaccine. Okay, we're going to say that 0 stands for placebo and 1 stands for vaccine. It's going to make everything easy from now on if we do that. Use the function sample to draw a random sample of size 196 from this cohort. Then use the function sum to compute how many people in the sample receive the vaccine. Quarkus use seed 123. Okay, so things suddenly got very dark. Now we have plenty of information to deal with, but we have everything we need. We just need to go step by step. Anybody has an idea how I start to answer that question? So let's read it again. We assign the number 0 to patients who receive the placebo and number 1 to patients who receive the vaccine. Okay, that's the easy part. 0 placebo, 1 vaccine. Okay. And we want to draw 196 people. Like they're not really people, they're numbers, from a total of 30,420. Okay, someone in the chat proposes a solution, which is sample rep equals x equals 0.1. Oh, it's a very long solution. Okay, so we're going to look at the answer that's in the chat. Okay, I'm going to type it so that everybody can see it, even people in the room. So I'm going to type it step by step. And you guys will tell me what you think about it. Oh, okay, so that's part of the answer here. That's what is suggested. Okay, sample x C01 size 196 replace equals true, and wrap it all up in sum here, like that. So that's what's on the chat. That's the solution that's on the chat. But we're going to go step by step. That's important to so that we don't go too fast. Let's first discuss that particular command here. So there's a question, sorry, will the recording be available as soon as this is over? My internet is disconnecting and reconnecting me. Yeah, it will be, I'll put it on YouTube if it's okay with everybody. If there are some people who have issues with this, then I can put it as a Zoom video only shared with the people in the course. But I think on YouTube, it's an easier solution for everybody. It takes me about 1 to 2 hours to make the video for YouTube. It's not a lot of work, it's the time it takes to convert the video. So it's going to be available from this evening. Okay, so let this thing here. What does this do? Any idea? I can actually press enter and see what this does. Okay, so comment anyone. What does this function, this command do here? I'm sorry, I can't hear anything. Yeah, it's randomly assigning either a 0 or a 1 to 196 people, okay? So if I don't do replace equals true here, what's going to happen? Well, I get the error that I demonstrated in the video, okay? And so the reason is because you draw 0 and then 1, and out of numbers, you can't continue. So you need to have replace equals true here, okay? Otherwise, you won't be able to pull 196 numbers out of just 2, 0 and 1. Okay, and wrapping it all up in sum here, what does this do? When I put this sum like that, you see I get these things here. What does the sum do in that case? Any idea? Yeah, sorry. Right, exactly, okay? If you have only zeros and ones, when you compute the sum, you count the ones. Right? So, and the ones are the people who have received the vaccine, that's what we said. Okay, so when we do sum like that, it means that you get the number of people who have received the vaccine. Okay, so what you're doing in this sense, what you're simulating or what you want simulates. So yeah, the answer in the chat adds all the ones. Exactly, it's counting the ones if you add them. So what we're doing in this setup is that we're saying, okay, I have plenty of people who have either labeled 0 or labeled 1. They have received the vaccine or the placebo. I'm getting 196 at random. How many of them have received the vaccine? And the first time I got 105, second time I got 98, second 97, etc. It's clear for everybody, right, that I won't get always the same number. This is random. So if I get 196, if there are equal amount of zeros and ones, I should get approximately half, but not exactly half. It's going to be different, right? So what I'm doing now is to get a feel for how far and how much can I get, how high can that be, and how low can that be, okay? And so if I start to play around a little bit by just repeating the command again, I can see that the highest number I got was 105, 113 even, okay? And the lowest I got was what, 84. Okay, so these are just purely random fluctuations. It means that if I just have equal amount of people with 0 and 1, and then I just grab 196 of them, right, I could get anywhere between 84 and 113 who have the number 1 on them. Okay, there's a range of fluctuation. And so that's why I need to understand that range of fluctuation. Did I ever get 5 or 179? No, and probably I won't. Okay, so what I'm trying to do here is to just get a feel for how much variation there is and how different the numbers can be from each other. Okay, so that's in a sense what happens in this call. So is that really the best way to answer the question? Well, you could say yes, but you could do a little bit better. Okay, so in a sense, what if I want to really represent the fact that there were 30,420 people and we've drawn them without replacement. Okay, like what I'm doing here is not exactly the same to say there's an infinite number of people and I'm replacing them, or to say, no, there's a finite number of people, there are 30,420, and when I pick someone, I pick them only once, I cannot pick them twice. Okay, so here, how would I actually make this with replace equals false? Okay, so I'm going to like emphasize that I need that here. So here that doesn't work. So what should I put here on the X to represent the fact that we have 30,420 people who volunteer? So it's almost exactly the same, but not exactly the same as having infinite number of people and then drawing 196, or having 30,000 and drawing 196. Which function am I going to use now? The one that we've just seen before. Any idea? To produce a vector of size 30,420. Rep. Someone in the chat says rep. Indeed. Okay. So here I want to repeat that thing here. So I want to repeat 0, 1 times equals 15,210. Okay, and that works. Right? Of course, the samples are indistinguishable. It's just a bunch of zeros and ones. But internally, this is not exactly the same thing. To say I generate now a finite pool of 30,000 volunteers, each with label 1 and 0. Okay, and from there, I take 196, but I pick each person only once, and I can't pick the same person twice. Okay, so that thing here is an enormous monster. Okay, here's going to be 0, 1, 0, 1, 0, 1, 0, 1, repeated 30,000 times. Okay, if you don't trust me, do it on your session here, but if I do that, it's going to fill the screen with plenty of zeros and ones, and we're not going to be able to to see what's up there. So I'm not going to press enter, but that vector here is a huge vector of size 30,000 with alternating zeros and ones. Okay. Right. Does that make sense for everybody that this second way of solving the problem is a better representation of the trial? Okay, it doesn't assume an infinite number of people who are drawn multiple times, or just two people who are drawn multiple times, sorry. It really says they were 30,000 volunteers, so we model them all, we put them all inside the box, and we draw each one of them, but only once. Okay? Online that seems to be okay as well. So let's answer the question then. So it says use set seed 1 to 3 as usual. So here we go. Set seed 1 to 3. Okay, that's the random sample that we're going to use here, and we sum to count the number of ones. So that's 97. So the answer on Quarkus is 97. Okay, question, comment so far? Online, any question or comment? So this is why we need the function sample. Okay, we want to be, if we want to understand what happened in this clinical trial, we need to put the elements in the random generator, model it, and then see what would happen by chance, just to check whether what happened can be explained by chance or cannot be explained by chance. So randomized clinical trials are perfect examples of statistical tests. A statistical test is a protocol consisting of 5 things: a null hypothesis, an alternative hypothesis, a protocol to collect data, a test statistic, not statistics, it's a statistic, it's a value, and a decision rule. So whenever you think about having a quantitative approach, you always need to think about these 5 things. That's the golden rule of quantitative thinking. Okay, it's not just quantitative biology. It's like every problem that you're going to encounter when you want to approach it rationally and there's a random component, you need to know this method with the 5 things. Question 10. What are the null and the alternative hypothesis in the case of the Moderna clinical trial? So there's no reason you should know that, but we can guess. So who has a guess about what the null hypothesis could be in a clinical trial? Go ahead. Yes, the null hypothesis would be that there is no significant difference. Okay, more specifically, that whether you receive the placebo or the vaccine, then it doesn't change anything. Okay, so that's what the null hypothesis is that there is no that the treatment is exactly identical to the placebo. The treatment is a placebo. That's the null hypothesis. So if it's the null hypothesis, what would be the alternative hypothesis? Can you speak a bit louder? I can hear. Okay, the alternative hypothesis would be that there would be statistically, you'd feel statistically better, you'd have less infection, etc., if you have received the vaccine than if you have received the placebo. That's what you said, right? Yes. So it's this, almost this, okay? Is that there is no notion of statistical significance in the alternative hypothesis. And this is why this method in 5 points is really important. That will be much later, like 1 and 2 is really what is truth, without thinking at all whether this is measured, measurable, or something like this. Okay, so think about it, steps 1 and 2, as without notion of randomness. Either the vaccine is as efficient as the placebo, or it's more efficient than the placebo. No discussion yet about whether this is, is going to be significantly better or statistically better or something like this. Okay, so we have only 2 cases in our mind. Either it's precisely exactly identical to the placebo, not approximately equal, exactly like the placebo, or it's going to be better. Okay, there's another alternative hypothesis, which we could say it can be worse as well. Okay, that's a sense in which the null hypothesis would not be true. And so we have to kind of make up our mind a little bit about whether we want to include this alternative hypothesis. And in general, people do. They say, if you don't know, why not include it as a possibility after all? It is possible that after you receive the vaccine, you get more infections than with placebo. After all, why not? And mind you, this is what some people have reported for the third dose or something like this. They have said that the 2 weeks after the vaccine, there was a bit of a drop in the quality of your immune system. So after all, why not? So I would suggest that the null hypothesis would be the vaccine is exactly as good as the placebo, and the alternative would be it's better or worse, but not exactly as good. Okay? And that here is actually quite an important step, even though that seems completely trite, because I insist that there is no notion of randomness. We're just describing the possibility the world can be without notion yet of randomness. Randomness is going to come later. Question 11. What is the protocol to collect data in the case of Moderna clinical trial? Any idea? It's actually what I read. So that's the randomized clinical trial is a method to collect data. That's exactly what it is. Okay, and this method includes the blindness and the randomization. So it's a randomized observer-blind clinical trial. That's the method to collect the data because it specifies what you're going to do in order to build your data set, as simple as that. Okay, there's much more to it than just that because they had to consider questions about the health level of people in the beginning, etc. But they decided this beforehand. Okay, they did not wait for people to tell you, "Oh no, I don't like you, just go away." Like, no, we're not giving. You're not part of the trial. So they had very strict establishment of who can and who cannot participate to the clinical trial, and that's what constitutes the method, the protocol to collect data. So in a sense, that is the whole randomized clinical trial. And the answer is it's this randomized clinical trial with observer blind protocol. Okay, so this is the part that is extremely important. But that's the one we're going to leave to the experts because that's really something that is dedicated for clinical trials. And I can barely comment on this. This is not my expertise at all. Question 12, propose a score that is expected to have a different value depending on whether the null or the alternative hypothesis is true. Okay, and this here is the key. That is the key of what we're doing of statistic. Basically, it's question 12. That's the one that is number 4 here. Okay, you need to choose a good score that makes a difference between null hypothesis and the alternative hypothesis. So go ahead, suggestions. What could we use as a score that would tell us whether it's more likely that the null hypothesis is true or more likely that the alternative hypothesis is true? Any ideas? %95. Okay, so using the 95% threshold. Very good. Other suggestions? Let's get all the ideas now. Something like p-value. Okay, the number of participants who develop symptomatic COVID-19s among the vaccinated. Okay, other ideas? So the first thing is, what do we mean by a statistic? What is it? What do we mean by a score? Okay, and so what is the most important thing of a score? Well, the first thing is that you have to be able to compute it. Okay, so whenever you choose a score for something like that, so the very first thing that you must ask yourself is, can I compute it or not? And that's what defines a statistic versus a score. A statistic is something that if you look up the definition in the statistics book, it's a number that you can compute all the time. Okay, whatever happens, you know that you will be able to compute this score. So why would you not be able to compute a score? Because it depends on some unknown parameters. Okay, so for example, if you're thinking of measuring something about the weather, and like you're wondering about the increase in temperature in the Earth, if your score depends on the temperature of the Earth in 2100, you can't compute it. This is unknown. So you can have it on a piece of paper to say my score depends on that, but you can't compute it. Okay, you can define it, but you can't compute it. The statistic, all it means, it's a score that you can compute all the time. So it does not depend on any unknown parameter. Okay, so that's what we mean here. So we had the score at 95%, threshold 95%, the p-value, a relatively long answer, the number of participants who develop symptomatic COVID-19 among the vaccinated. Okay, so can we compute the p-value? I guess we can, but not right now. We're not good enough. So this one, let's put this on this side. At some point we'll use it, but in terms of computing, this one is not going to go through just yet. The other suggestion was a threshold of 95%. Again, so excellent idea. The issue is that we're not yet able to compute something like that. So we want something much more concrete. And so the main proposal we have yet is the number of participants who develop symptomatic COVID-19 among the vaccinated. Is that a good score? Is that a bad score? What are your ideas around here? Does anybody think it's a bad idea to use that score? That there's a problem with it? No? Does anybody think it's a good idea except the person who proposed it? So the opinions are kind of a little bit mixed. So last question about this, what a good statistic is. Does it make a difference? Okay, do you expect the number to be different based on the null hypothesis versus the alternative hypothesis? Okay, so do you expect that if the null hypothesis is true or the alternative hypothesis is true, the number of people who have COVID-19 among the vaccinated is going to be different, or is it going to be the same? Yeah, it's going to be different, right? We have this idea that if the alternative hypothesis is true, then the vaccine is more efficient than the placebo, or maybe less efficient. But either way, it means that among the people who get the vaccine, okay, you will get not the same number of symptomatic COVID-19. All right, so is this a good score? Well, it seems like there's an idea. I would argue against that, okay? I don't like that score completely. I like it a lot, but not completely. Why is that? Is because if we just say there are 15,000 people who got the vaccine, let's count how many of them get COVID-19. What about the placebo? Well, like, did we just vaccinate them for the fun and then we're throwing them away? Like, it seems that we're not really using the placebo at all if we take that score. So in a sense, you could say, oh my God, there are like only 7 people among the vaccinated who got COVID-19. Yeah, but in placebo there were 3, for instance. Well, like the vaccine seems to be worse than the placebo, or maybe equally good or equally bad. So what else, almost like that, what else could we use as a score? So one idea is to say we compute among the people who get COVID-19, how many are vaccinated. Okay, that takes into account the placebo this time, right? So it's not at all the same to say, let's focus on the 15,000 people who got the vaccine and see how many people have got the disease. No, we say, okay, at the end of the clinical trial, after a certain amount of time, let's round up all the people who got COVID-19, who got symptomatic COVID-19, like they got it bad. And then we see how many of them got the vaccine. Okay? So this seems much better because those who did not get the vaccine got the placebo in that case, for sure. And so let's say we have 1,000 people who got COVID-19, and then we open their status and we find that 500 have got the vaccine. What does that tell you about the hypothesis? Is it going to be the null or the alternative? The null in that case. Like if it's 50-50, then it means that if 500 have got the vaccine, 500 have got the placebo, they're equally efficient. And if you see that among this 1,000, 2 have got the vaccine, well, it means that 998 have got the placebo, so the null hypothesis is probably false. The alternative is probably correct. Okay, so that's the score we're going to use. Right, so there were other, while I was talking, there were other suggestions. They're really good in the chat. The total number of participants who developed symptomatic COVID-19 in the trial, then maybe the ratio. So the idea is kind of close to the ratio, but in the end, that's the score we use. It's take the people who got COVID-19, out of those, count how many have got the vaccine. So imagine that there were 196 people who got COVID-19 in this trial. Okay, if the placebo is as good as the vaccine, then we expect half of this 196 to have the vaccine and half to have the placebo. Right? Does that make sense for everybody? Right. So a lot of the details matter. Sorry that this discussion takes long, but this is really the key. Like this step is always the key to the statistical analysis. So we're going to use that score. It doesn't really have a name, but that's the number of people who have the vaccine among those who got COVID-19, and not the other way around. We now have to find a good decision rule. In order to do this, we need to understand how the score behaves when the null hypothesis is true. This is when we use randomization in order to get an idea of what to expect if the clinical trial fails. If the clinical trial fails, it means that the placebo is exactly as good as the vaccine. All right, so let's go back to this list here because that's the most important. So we got a null hypothesis. We got an alternative hypothesis. These are non-random. Okay, we got a way in which the data was collected. We just trusted, still not random. Now the most important step is number 4. We choose a score that really discriminates between the null hypothesis and the alternative hypothesis. And now we need to make a decision rule which will tell us what are we going to decide depending on what we observe. And for this, we need to understand how our statistic behaves. And this is where we need randomization. Repeat 5 times the procedure of question 9 and record the lowest value. Okay, so question 9, what was it? Is the one that's still here. Okay. And we need to record the lowest value. So the first was 97. Okay, and we're going to repeat it 4 times from here. Okay, so we got 5 numbers, and you can see that I've used set seed here just once, and from that moment, then I reset the state of the generator, and I'm moving forward. I'm getting random samples. And if I repeat the whole process like that, 1, 2, 3, 4 time, you can see that the first 5 times and the second 5 times, they're identical. Okay, so once I set the random number generator, even, doesn't matter how many steps I make, like I fixed it and I will observe exactly the same result every time. So that's what it means here. Use seed 1, 2, 3 once and run the command 5 times. Okay, that guarantees that you always get the same 5 results. So among those, what is the lowest value? It's 85. Okay, so to recap, what am I doing here? I'm saying that I'm going to look among the people who got COVID-19, how many have got the vaccine. Okay, that number, if it's going to be very low, like 0, I'm going to conclude, wow, it's just super efficient because all the people who got COVID-19 got the placebo. And the question is, how low will be the threshold to tell me, yeah, okay, that's not random. Okay, so I'm just going to look at how low I get the numbers when I just resample things at random, imagining that the vaccine is inefficient. Imagine I've got the same chance of drawing a person with a placebo or with a vaccine, and let's see how low we can go. And we see that 85, here it's a normal low. Okay, if we got 85 people out of 196 who got the vaccine, I'd say, it's random. Doesn't look like this is particularly low. Like even out of just 5 times, I got something like this. Okay, does this argument make sense to you? To say like, I tried 5 times with the null hypothesis, I got 85. So if you come with 85, I say it's not much better than random. Does that make sense? Right, that randomness produces the same numbers as you, basically. So like, it just looks like I take my null hypothesis, I randomize things, I get 85. So if you got 85 in the clinical trial, then I'm not impressed. because I got it by chance, even when the vaccine is not very efficient. Okay, that's the whole logic of this approach. Is compare what you obtain by chance and see if the real data is better, more impressive than what you obtain by chance. Okay. All right, let's move on. Where were we? The commands become more and more complicated. It is time to introduce a way to simplify them by defining a variable. A variable can hold the result of any operation in R. It is defined with the operator less than and a dash, a minus. Below is an example showing how to define a variable called cohort. Okay, so it's this command that's here, cohort, and I'm here defining the word cohort. It's not existing in R before you define it this way. And I'm saying that cohort is this humongous vector of size 30,000 with 0 ones that alternate. And I call it the cohort because that represents the volunteers as they arrived at the clinical trial. Okay, 0, 1, 0, 1, they were all alternating placebo vaccine, placebo vaccine. Okay, so I call this cohort, and that vector now holds the 30,000 values. Okay, so that's a much more compact way of doing it. So it's just because it's more compact and makes everything more readable. You think of the cohort as the cohort instead of this unreadable rep x c01, etc. This vector will come in handy to answer some questions. We can see cohort as a shortcut for rep x equals c01 times equals 1, 5 to 10. The question below shows how this can simplify the commands. Okay, so question 14 answer question 9 using the vector cohort as defined above. So it's this call here, the call of question 9. Sorry. All right, so that's this horribly complicated thing, but I say that here I can just replace that by cohort, and I get the same result. Okay, so just to show it. That was the same 97 as we observed before, because here cohort is just a shortcut for what we had before. Okay, and that makes it a little bit more readable to say sample from the cohort, 196 people. All right, so we're getting there. Our procedure so far provided some typical values of the statistic when the null hypothesis is true. That is to say, when the vaccine is no better than the placebo. If the number of vaccinated patients with symptomatic COVID-19 were any of those typical values, anywhere between 85 and 113, there would be little evidence that the vaccine is effective. Okay, I repeat, this is really the essence of what we do. We randomize things. We say just by chance you expect anywhere between 85 and 113. So if you're within this window, then you're not better than chance. Okay, and we need to go through the whole process to do this. Otherwise, we'd not know where this window is. Okay. So to complete our decision rule, we need to find some numbers that are unlikely to occur when the null hypothesis is true. So we need to see what are the borders where we'll still consider that this is random, but if you give some numbers that are not in this window, I'll say, no, like these numbers that you're coming with now, they cannot be explained by chance. The only way they can be explained is that the null hypothesis is false. There has to be a non-random process that generates these numbers. The function replicate allows us to repeat a complex operation a large number of times. For instance, the command below produces 3 random samples of 2 numbers from 1 to 5 and computes the sum for each sample. Okay, so I'm going to decompose this step by step. So there is this sample here. Okay, I'm sampling 2 numbers out of 5. I'm computing the sum here. So we've seen this before in other contexts. And when I do replicate n equals 3, blah, 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 with the whole thing, it's a pretty long command now, you can see that I get 3 numbers because I've done the whole relatively complicated operation 3 times. Okay, take 2 numbers, sum them. Okay, that's 3 here. I must have gotten 1 and 2. Then repeat again. Take 2 numbers, sum them, get 7, etc. Okay, so this replicate n equals something. It's a way to reproduce a relatively complex operation a large number of times so that we can see rare events now. This will be useful to produce rare events with frequency of 1 in 100 or less, in turn giving a threshold for the decision rules of statistical tests. So question 15, use the function replicate to repeat 1,000 times the procedure of question 9 and use the function min to output the minimum. Okay, so just plug and play here. We're not trying to try and understand very much how this all works, but here this is the command, that computes how many people are vaccinated in a sample of 196. So what does this I want? Replicate it 1,000 times. Okay, so replicate n equals, let's say, 5, 4. Start. And then I said the expression is the whole complicated thing I had before. And you can see I get 4 numbers in one go. Okay, that's just a way to repeat the whole thing instead of pressing up arrow a million times. I put this inside the replicate. I can increase it to get a little bit more throughput. You can see that now I start to get a better idea of how things look by chance. Okay, if I could reproduce the randomized clinical trial 100 times and the placebo and the vaccine were as efficient, I'd get these numbers. Okay, so now the question says do it 1,000 times. Well, we can press enter and then we'll see 1,000 numbers. We want to know the minimum. So we wrap it all in the function min. That's a pretty complicated case, but in the end, it all makes sense, and it's 77. So what does that mean? It means that if we could 1,000 times replicate the randomized clinical trial and the vaccine was doing nothing, the lowest number we see among the people who got COVID-19 is 77. If you get something lower than that, it's not really explainable by chance alone. Okay, so the question says use seed 1 to 3 as usual. So just to make sure. Okay, it's the answer on Quarkus is 79. Okay, so out of 1,000 trials, the chance that you'd see something much lower than this is very, very small. Okay, so if people at the clinical trial come up with a number that's above 79, when they compute the number of people who got the vaccine in the COVID-19 cohort, then I'd say it's random. Like it's maybe not random, but randomness can explain this. Okay, it's only one chance in 1,000. I know that, but still, it's like chance can somehow explain that. So if the null hypothesis is true, that is to say, if the vaccine is exactly as efficient as the placebo, the number of vaccinated participants among the 196 who develop symptomatic COVID-19 cannot be too low. In fact, the chance is less than 1 in 1,000, approximately, that we observe a number that is smaller than the minimum obtained at question 15. Okay, so we have only approximately 1 in 1,000 chance to obtain a number lower than 79, just out of random. We can thus use this value as a threshold. If the number of vaccinated patients among the 196 with COVID-19 is lower than this value, we will have to conclude that chance alone is not a plausible explanation for the result. In that case, we will reject the null hypothesis and conclude that the vaccine is more efficient than the placebo. Otherwise, there will be insufficient evidence to claim that the vaccine is more efficient than the placebo, and we will accept the null hypothesis. Okay, question 16. In the Moderna clinical trial, 11 out of 196 patients with COVID-19, symptomatic COVID-19, were vaccinated. Conclude. So what do you think? I've after all this preliminary trial work that we did, the answer is 11. Now what do you guys say? Does the vaccine work? Does it not work? Okay, so we have this window, which is the random window. And we say that approximately starting 79 and up, okay, this can be explained by chance. If it's much below 79, it's very unlikely that chance alone can explain the result. They got 11, okay? Which means that this is much, much lower than 79, much, much lower. There's no chance that you would get 11 people only out of 196 who got the vaccine if the vaccine is as efficient as the placebo. It's way, way too low. Any number way below 79 is not compatible with the null hypothesis. So someone says in the chat, reject the null hypothesis. Yes, we have to, okay? Like the numbers that we observe are incompatible with the predictions of the null hypothesis. The null hypothesis says you should get anywhere between 79 and whatever, 120, 130, something like this. We got 11, it's completely out of the window. We said the null hypothesis has to be false. We take the alternative hypothesis instead. And the alternative hypothesis is that the vaccine is not equal to the placebo because there are much fewer people who have COVID-19 when they have the vaccine, then we conclude that the vaccine is more efficient than the placebo, not less efficient. Okay, so let's recap here. The method is that we need to think about what are the possible cases, okay, and regardless of randomness, etc. Like what can the world be? Either the vaccine is as efficient as the placebo, or it's not as efficient, it's more efficient. Okay, once we got this, we choose a score that's supposed to be different depending on whether hypothesis A or B is true. Then we say, when hypothesis A is true, how much is going to vary that score? Oh, it's going to be between this and this, between these two numbers. And then we look, in reality, is it within this window? If it is, we say, well, yeah, I thought that with hypothesis A, we'd get some, of course, there will be some randomness, and it's going to be between these two values, and it's right in the middle, so it's compatible, it's fine. But if the number is way there, way out of my window, then I say, whatever, my hypothesis, even when I stretch it to the extreme, to 1 chance in 1,000, still not able to account for the real that has happened. So I've got to throw away my hypothesis, right? It doesn't work, my hypothesis. I need to change my mind. I need to say whatever I believed about this hypothesis, it's not a good one. It doesn't correspond to the reality at all. And what the whole process allows me to do is just to incorporate the fact that there is some randomness in the process. That's why I need to do the whole thing with a sample. Okay? All right, questions or comments or things you'd like me to repeat, to go over once more about this whole process? Please have the number been 78 or 77, would the answer be the same? Because our cutoff is 79. Okay, if we want to be strict, we say 79 is the deadline. If you're 79 or more, we consider that this is probably compatible with random. If it's below, they say no, we reject the null hypothesis. This cannot be random. So in that case, we'd be borderline, but we still be rejecting the null hypothesis. Here we are not borderline at all. We're way below the minimum possible observed by chance. Okay, so it's always uncomfortable when you have to make decisions that are borderline, but here it's not the case. When it is the case, we're strict. We say below the cutoff, reject, above the cutoff or equal, accept. Okay, so if it feels hard for you to understand, it's good. This is a really hard process. Okay, if this class feels really hard, it's because it is the hardest. Okay, in the sense that the level of the classes, the level of difficulty is going to increase, but this is the class where your knowledge and the target knowledge is the gap is the biggest. The level of the classes is going to increase, but your level is going to increase much faster. So it's completely okay if you're lost. I think that it'd be even worrisome if you're not lost. It means that the course would be too easy for you. these concepts are all new for you. Okay, so I know that I'm throwing a lot of answers at your face. I think we're going to stop here. There's not a lot of time for the other things. You still have 5 questions to answer. They're pretty difficult, but you've got all the keys already. Like we've discussed all the essential steps. It's more of the same. But what I want to insist on is the fact that really this class has got so much new elements for you. Okay, some of you know statistics. I've heard the words p-value, threshold, etc. Forget all of it. Okay? Just try to think about what does it mean to be random? What does it mean to make a decision in the presence of randomness? Can we even make a rational decision in the presence of randomness? What does that look like? And I'm proposing a protocol. That's of course the protocol that science proposes. There's just an idea, there's just a solution. And this thing is so counterintuitive that it'd be really weird if you did not have resistance to these concepts the first time. So maybe it's going to feel, it's going to hurt the first time. Maybe you're even going to, panic a little bit at the moment of filling in the answers. If you do, don't worry. Okay, that class is the hardest in that sense, that you're the difference between where you are now and where you need to be is the biggest, but you will really rapidly catch up. You'll see we'll do every time the same thing. Is going to be the same protocol, the same ideas again, just we're going to see them slightly different variations, and these ideas are going to sink in. The most important for you, if you're interested in, you can drop the course, of course. Like, this is, you can say, no, nothing's for me, etc. That's not what I like. If you're interested, if you want to continue with the course, great, I'll help you, of course, and you'll get there. The most important thing is that you try to understand, why does that work? How does that work? Don't try to just remember the steps. I'll never ask you questions of memory, ever. I just want you to understand what's the big idea here. Because there are some pretty big ideas there, and it takes time to wrap your head around it. Okay, so in the last 3 minutes, I'll take more questions, or comments, or things that you'd like to discuss. But otherwise, we'll stop here, and then you'll have to finish from 7 till the end. There are some additional questions if you're really hardcore and you want to go like read till the end. Questions from 21 or for bonus points. So they're optional. If you feel like trying to answer them, you never know, maybe you're going to get some extra points, extra marks. Otherwise, I'll be happy to answer any remaining questions or discuss anything. Okay, no questions in the chat, no questions here. Very good, so that's a wrap. Thank you very much for your attention, and good luck with the quiz, good luck with the assignment.